Spermatogenesis is a process. It's a series of events that give rise to sperm cells. And each time there's an event, a cell division, the cell takes on a new name. So let me show you what I mean. On the outer periphery of the seminiferous tubule are the very earliest spermatogenic cells. They're called spermatogonia, and spermatogonia only exist on the outer periphery. A male is born with spermatogonia, and they stay quiet until puberty. But at the time of puberty, hormones are going to stimulate spermatogonia to do two things. One is to undergo mitosis. And the reason that spermatogonia undergo mitosis is so that we don't have a declining number. So in other words, this is regular cell division. One spermatogonium gives rise to two spermatogonia. And then we can think of it in this way. Spermatogonia divide by mitosis to keep a bank of spermatogonia available. And so in this picture, we can see that one is going to stay in the bank, so to speak, and then one is going to enter into meiosis. So that one spermatogonium that enters into prophase 1 and starts the crossing over events and all those nuclear events that we see, that one cell is known as a primary spermatocyte. Even though it hasn't divided once, the primary spermatocyte is moved past the blood-testis barrier into the adluminal compartment, and that primary spermatocyte will undergo the first meiotic division, known as the reduction division, giving rise to two cells, secondary spermatocytes. Those two cells will each have 23 full chromosomes, or 23 chromosomes that have replicated DNA. So the two secondary spermatocytes will undergo the second meiotic division, giving spermatids. The spermatids are going to have 23 chromatids each. The spermatids are genetically sperm cells. They just have to lose this excess cytoplasm and grow the tail, the flagellum, and that's a process called spermiogenesis that I'll look at in a different video. But this is the process of spermatogenesis. Those early spermatids, like we said, will undergo spermiogenesis, giving rise to late spermatids and spermatozoa. Or to look at this in another way, here's a small segment of the seminiferous tubule. This is the basement membrane. This is the outer periphery of the seminiferous tubule. And we can see two pink sustentacular cells. And then the spermatogenic cells that are on the outer periphery are spermatogonia. Once a spermatogonium enters into meiosis, it gets pushed past the blood-testis barrier, and it's then called a primary spermatocyte. That primary spermatocyte will divide to give rise to two secondary spermatocytes, and then the secondary spermatocytes divide to give rise to spermatids, early spermatids, and then late spermatids.